Hello, everyone. Welcome to BioTuring Webinar on the topography of spatial gene expression in the human prefrontal cortex. This is Jenny from BioTuring, and today I will be the moderator of our discussion. Okay, and first of all, I'm super excited to introduce our speakers of today, Dr. Kristen Maynard and Dr. Maynard Collado-Torres. So about Dr. Kristen Maynard, Kristen is currently the research scientist at Lieber Institute for Brain Development, and she's currently studying the links between gene expression, plasticity, and the behavior in the context of psychiatric disease. Kristen has also spearheaded Lieber's efforts into dissecting gene expression across finer resolutions in the human brain. Dr. Leonardo Collado-Torres is currently the investigator at Lieber Institute for Brain Development. And Leo is also maintaining several bioconductor packages, including Spatial Lieber for spatial transcriptomics data. He's also the co-founder of the Lieber R-Stats Club, the CDSB Mexico community of R and bioconductor developers in Latin America. Also, Leo is serving on the Bioconductor Community Advisory Board. So for today's webinar, Kristen and Leo will be sharing with you guys their exciting work published on Nature Neuroscience using 10x Visium technology to characterize the human dorsolateral prefrontal cortex. They will be sharing about the study design and the data analysis parts. And also in the later part of the talk, we will have a 10-minute tutorial with our head of software development, Trile. He will be sharing with you guys how you can interactively explore this multi-slide spatial transcriptomics dataset in vBrowser, a platform for single-cell data analysis, as well as for spatial transcriptomics data analysis. And remember that you guys have 10 minutes at the end of the webinar for a Q&A section, so please stay tuned. If you have any questions along the way during the talk, then feel free to leave them in the Q&A box, which is located right below your screen. And we will go through them one by one in the Q&A section after that on a first-come, first-served basis. And if you would like to directly speak with the presenters, feel free to raise your hand during the Q&A section so that we will unmute you. Okay, let's get started. Kristen, you can share the screen now.
Okay, thanks, Jenny, for the kind introduction, and thanks to BioTuring for the invitation to speak, and to all of you for tuning in. Leo and I are excited for this opportunity to share our recent work exploring spatial gene expression in the human dorsolateral prefrontal cortex using Visium technology. A remarkable aspect of the human brain is its highly organized structure-function relationships, especially in the cerebral cortex. So the cortex plays a key role in many functions, including perception, awareness, thought, memory, language, and movement, to name a few, and specialized cortical areas subserve each of these functions. The dorsolateral prefrontal cortex, which I'll be referring to as the DLPFC, is especially important for executive functions, including working memory, cognitive flexibility, and planning. And disruptions of this brain region have been associated with several psychiatric and neurodevelopmental disorders, including schizophrenia and autism spectrum disorder. The spatial organization of the neocortex reflects its complex function. A defining architectural feature of the cortex is its subdivision into 6 layers, each containing a unique distribution of different cell types and connections with other cortical or subcortical brain regions. In the cortex especially, the position of a cell within a specific layer influences its gene expression, morphology, physiology, and function. And indeed, studies in the postmortem brains of individuals with schizophrenia and autism spectrum disorder have implicated not only specific cell types in these conditions, but also reveal differences in neuronal and synaptic structure that are spatially localized to specific cortical layers. So in the past few years, there have been several rapidly evolving transcriptomic approaches for studying gene expression in the brain using RNA sequencing. I like this clever Lego brain schematic, which illustrates how we've expanded from bulk RNA sequencing, which contains a mixture of cell types, to single cell or single nucleus RNA sequencing, which allows us to understand molecular profiles for different cell types, and then most recently to spatial transcriptomics, which aims to link gene expression to its anatomical location to add molecular insights into structure-function relationships. And in fact, spatially resolved transcriptomics is a rapidly evolving field that was actually just named Method of the Year by Nature Methods for its power to unlock relationships between gene expression and the spatial organization of complex tissues. But taken together, these approaches really provide a powerful toolbox for investigating gene expression in the brain. So to further our understanding of gene expression within the context of the spatial organization of the human cortex, we use the recently released 10x Genomics Visium platform to generate spatial maps of gene expression in the 6-layer DLPFC of the adult human brain. Visium uses a patterned array of spots with barcoded oligo-DT primers to capture the mRNA from a tissue section. Then during cDNA synthesis, which occurs on the surface of the slide, barcodes indicating the location of each spot are incorporated into the cDNA, which is then removed from the slide, prepared into libraries, and sequenced. And this basically allows each read to be mapped back to the correct spatial coordinates on a histological image. Resolution is limited only by the spot size and the spacing on the array. And the current platform, each spot is 55 microns in diameter and spaced 100 microns center to center. We also use the 10x Genomics single-cell RNA sequencing technology to identify molecularly defined cell types in human DLPFC. This droplet-based approach uses uniquely barcoded gel beads, which are mixed with nuclei and added to an oil surfactant solution to create these single-cell gel beads in emulsion, or GEMs, which act as an individual reaction vesicle in which cDNA for that nucleus is barcoded. And this takes place on a piece of equipment called the chromium controller. Barcoded products are then pooled for downstream reactions to create sequencing libraries. And after sequencing, the unique barcodes are used to map reads back to their original single nucleus of origin. And I just want to point out that while single-cell RNA-seq approaches are common in rodent, tissue, and cultured cells, the fact that most available post-mortem human brain tissue is frozen has resulted in the majority of data in the human brain being generated on isolated nuclei with single nuclei RNA-seq approaches. So to give a brief overview of the scope of the webinar, I'll be describing how we use Visium to identify layer-enriched genes in human cortex and how we validated these findings using in situ hybridization. And then I'll pass off to Leo, who will talk about spatial registration of single nucleus RNA sequencing data and laminar enrichment of genes associated with different brain disorders. Before we dive into the study design and data, I'd also like to acknowledge some of the people who significantly contributed to this work, especially our collaborators at 10x Genomics, Stephanie Hicks and Lucas Weber at the Johns Hopkins Bloomberg School of Public Health, and Carrie Martinovich and Andrew Jaffe at the Lieber Institute. This was a team effort, and we really had a lot of fun working together on this project. So in our study, we profiled human postmortem DLPFC tissue sections from 2 pairs of spatial replicates from 3 independent neurotypical adult donors. And each pair consisted of 2 directly adjacent 10-micron serial tissue sections, with a second pair located 300 microns posterior from the first, so resulting in a total of 12 samples run on the Visium platform. The tissue sections were acquired in the plane perpendicular to the peel surface that extended to the gray-white matter junction and included the 6 cortical layers as well as white matter. We sequenced each sample to a median depth of 300 million reads, which corresponded to a mean of approximately 3,500 unique molecular indices and a mean of approximately 1,700 genes per spot. The orientation of each sample was confirmed using marker genes for gray matter containing neurons, white matter containing glial cells, and previously identified laminar marker genes. So what you're looking at on the top panel is a DLPFC tissue block and corresponding histology from a representative section. The bottom panel shows spot plots depicting log transform normalized expression or log counts for SNAP25, MOBP, and PCP4. Expression of these genes confirmed the spatial orientation of each sample by delineating the border between gray matter, which is marked by SNAP25 enriched in neurons, and white matter marked by MOBP, or myelin-associated oligodendrocyte basic protein enriched in oligodendrocytes, and also defining layer 5, marked by PCP4, a protein important for synaptic plasticity. To give you a flavor of the collective data set, here are spot plots for PCP4 for all 12 tissue sections. There's one row per subject, and adjacent spatial replicates are shown in the columns. You can see there are slight differences in the dissections for each subject due to the folded nature of the cortex in the human brain. But we have the span of the cortical layers and the white matter, with cortical layer 5 enriched for PCP4. So using these data, we first generated aggregated layer-enriched expression profiles for each spatial replicate using a supervised approach. We use cytotectonic architecture and robustly expressed region or layer-enriched marker genes, combined with the dimensionality reduction method, specifically T-distributed stochastic neighbor embedding, or T-SNE, to assign individual spots to each of the 6 neocortical layers, or the white matter. Then we performed pseudo-bulking by summing the UMI counts for each gene within each layer across each spatial replicate to generate layer-enriched expression profiles. The pseudo-bulking approach summarized approximately 48,000 spots to 76 layer aggregated profiles across the 12 samples, thereby removing sparsity and greatly increasing the UMI coverage of genes. Principal component analysis of layer level or pseudo-bulked expression profiles across all sections and subjects revealed that the first principal component separates the white and the gray matter, and the second principal component associates with the different cortical layers. After pseudo-bulking, we used 3 strategies to perform differential expression analyses using these layer-enriched expression profiles with linear mixed effects modeling. So the first statistical model, termed the ANOVA model, involved testing for differences in mean expression across the 6 layers plus the white matter and asked, is any layer different? The second statistical model, termed the enrichment model, tested for differences in expression between one layer versus all other layers and asked, is one layer greater than the rest? The third statistical model, termed the pairwise model, tested for genes differentially expressed between each pair of layers, so 21 total pairs, and asked, is layer X greater than layer Y? You can see visual depictions of these 3 models using MOBP as an example. And remember, MOBP is a gene highly expressed in oligodendrocytes, which are enriched in the white matter. So using this approach, we systematically assessed the robustness of previously identified layer marker genes from rodent and human studies and our DLPFC layer-enriched gene expression data set. And we found that a subset of these canonical marker genes from the literature, about 60%, showed significant differential expression in our human DLPFC data, which were largely driven by markers identified in human cortex as opposed to rodent cortex. We also identified several genes previously underappreciated as laminar markers in human DLPFC. And here are a few example genes, including AQP4 enriched in layer 1, HPCal1 enriched in layer 2, TRAB-D2A enriched in layer 5, and KRT17 enriched in layer 6. Box plots and corresponding spot plots for these genes are depicted on the left. And then we validated these novel layer-enriched differentially expressed genes using multiplex single molecule fluorescent in situ hybridization. You can see AQP4 in red, HPCal1 in white, TRAB-D2A in pink, and KRT17 in green, with lipofusin tissue autofluorescence segregated into its own channel. So Visium is a powerful approach for detecting gradients of gene expression, as evident for KRT17, which shows increasing expression from superficial to deep cortical layers. One other aspect of the Visium platform that we've been harnessing to interpret spatial gene expression is the histological data. So we've developed image analysis methods for cell segmentation of the histology images to be able to estimate the number of cell bodies located in each 55 micron spot. So on the left, you can see an example of this segmentation in mouse brain, which has a higher density of cells in the cortex compared to human tissue. On the right, you can see high magnification images of the gray matter and the white matter of the human DLPFC. On average in DLPFC, we had 3.3 cells per spot, with a mean 15% of spots per sample containing a single cell body and 9.7% of spots containing no cell bodies. And below is a table depicting the breakdown by layer. We considered spots lacking cell bodies to be neuropil spots and predicted they would be enriched in dendrites and axons that form the connections or synapses between neurons. We were very interested in these synaptic neuropil spots, as this important anatomical information is lost in dissociated nuclear preparations commonly used for single nucleus RNA-seq studies. And indeed, using a previously published data set of genes expressed at rodent excitatory synapses from Hefner and colleagues in the Schumann lab, we identified significant enrichment of genes that are preferentially expressed in the transcriptome of synaptic terminals in our neuropil spots. And this is particularly relevant for brain disorders, as it's been shown that genes associated with increased risk for schizophrenia are preferentially enriched for synaptic neuropil, suggesting that this extranuclear information captured by Visium, but missed by single nucleus RNA-seq approaches, may be especially relevant for understanding genetic risk for brain disorders. So before I turn it over to Leo, I want to briefly introduce you to a new application of Visium that we've been working on called Visium immunofluorescence. Now, this flavor of Visium is particularly exciting because it allows for integration of proteomic and transcriptomic data. The workflow is exactly the same as I described before, but instead of doing H&E histology, we use fluorescent antibody staining for proteins that label different cell types, subcellular compartments, or even neuropathology. This immunofluorescence map is then directly integrated with transcriptomic data, enabling us to analyze relationships between gene and protein expression within the same exact tissue slice. So to give you a sense of one of our antibody panels, here is an example of immunostaining for 4 of the main cell types in the human inferior temporal cortex. NUN labeling neurons in green, TMEM119 labeling microglia in white, GFAP labeling astrocytes in yellow, and OLIG2 labeling oligodendrocytes in pink. So using this approach, we can now start to understand the cell types and the cellular compartments that contribute to the gene expression profile in each of these Visium spots. We're currently employing Visium IF in the human cortex to understand molecular and cellular changes associated with pathology and neurodegenerative disease. Okay, so now I'm going to turn it over to Leo, who's going to talk more about integration of Visium and single nucleus RNA sequencing data and clinical relevance of layer-enriched gene expression profiling.
Hey, thank you, Kristen. So I'm first going to explain how we can use our laminar data to perform a spatial registration of external single-cell or nuclei RNA-seq data sets. On panel A on the left, over here, we illustrate the process of taking your cells or nuclear clusters and assigning them to human DLPFC layers. The end result is a heat map, just like the one shown here on panel B on the right side, that shows the correlation between enrichment statistics computed on your data on the x-axis against those from our laminar data on the y-axis. The colors used for the correlation range from dark purple being -1 to dark green being +1. This specific heat map shows that our layers agree with the single nucleus RNA-seq data from Hodge et al. That's because we see this green diagonal over here. So let me explain this process more slowly. First, on your single cell or nucleic RNA-seq data, you have to identify clusters. Then you can pseudo-bulk your data and compute enrichment statistics, t-statistics, like we did for our spatial transcriptomics data. That is, you will find genes that have higher gene expression in a given cluster against other clusters, like illustrated over here. At this point, we have now two collections of enrichment t-statistics, one for our laminar data and one for your single cell or nucleic data. In order to make a heat map like the one on this slide, we first have to subset both collections of t-statistics to the genes that were tested in both data sets. We show our laminar layers on the y-axis and your clusters on the x-axis in a way where we hierarchically cluster the columns. In order to interpret the resulting heat map, we mostly focus on the dark green cells that present strong correlation between the enrichment t-statistics from both sets. This particular heat map shows the spatial registration for the Mathis et al. Dataset, where we see that all the oligodendrocyte clusters over here in the bottom left have high correlation with the white matter marker genes. Similarly, on the top left, we see that the astrocyte clusters have high correlation with the layer 1 marker genes. Then there's some excitatory neuron clusters, cluster 2, 0, 4, and 6, that have high correlation with layers 2 and 3 from our data. And then there's other excitatory neuron clusters from the Mathis et al. Dataset that have enrichment with either layer 4 or layer 5. So we can now differentiate the clusters of excitatory neurons across the DLPFC layers. We observe a similar story from our own DLPFC single nuclei data, and we can use now our laminar data to spatially register our clusters. Here, for example, all of the oligodendrocyte clusters on the bottom left have high correlation with the white matter marker genes. Like the astrocyte clusters have high correlation with layer 1, but then we also have excitatory neuron clusters, some of which have high correlation with, like, let's say, layer 2, layer 3, layer 4, etc. So in this way, we can annotate our single cell, or in our case, single nuclei clusters. And so these examples provide an illustration of how spatial registration can be used. And this particular last example uses data from a LIBD project led by Matthew Tran, who's a grad student in our team. Next, researchers have identified genes of interest through many different methods, and we can now ask if these genes are enriched in specific layers. Some projects create the literature, as is the case of the Safari project over here shown on the top. Others identify genes that increase the risk of a given disease or disorder through GWAS or TWAS methods, shown here on the left side. Or you can find differentially expressed genes between cases and controls, shown here on the bottom right. They all result in a set of gene IDs. So we can now ask if genes from these studies are enriched among the genes that show strong laminar enrichment effects in our data. One such case we examined was with genes associated to autism spectrum disorder, or ASD. The colors of the heat map shown here are on a -log10 p-value scale. Darker red boxes represent smaller p-values or higher -log10 p-values. We show that we only show the odd ratios for those cells over here, so these boxes over here, that have a -log10 p-value scale greater than 3. On the far left, we found that the Safari genes associated with, the Safari version 2, genes associated with autism spectrum disorder have strong odd ratios for layers 2, 5, and 6. But there's also a more recent study by, I'm hoping I'm pronouncing this correctly, Söderström et al. That is congruent with the Safari genes. This is a study from 2020. And they have 102 genes associated with ASD risk. And we find enrichment in layers 5 and 2. However, these 102 genes that they identified, they broke them up into 2 categories: ASD dominant traits and neurodevelopmental delay. And so we can see here that the 53 genes that they label as linked to ASD dominant traits are enriched for layer 5, and the 49 neurodevelopmental delay ones are associated with layer 2. So this suggested that we can assign the subclasses of neurons that might be contributing to the clinical outcome for each subgroup. This layer enrichment result for ASD risk genes was largely independent of ASD differential expressions from another study. If you want to do this type of layer enrichment gene assessment, you can do it through our own web application, spatial.libd.org/spatial-libd. You can also test genes associated with other disorders. Here we're testing on the right side genes associated with schizophrenia disorder. And in this particular case, we have results from 2 different studies, the PsychEncode study and the BrainSeq phase 2 study, where for each of these 2 studies, we have differentially expressed genes and TWAS associated genes. And so the differential expression results here are fairly concordant between the 2 studies, where we find genes that have higher expression in schizophrenia affected individuals are enriched for layers 1, 2, and 3 in both studies. The ones that have lower expression are somewhat congruent between the 2 studies, with white matter over here. There's some differences though. However, like if you compare the differential expression associated genes with the TWAS genes, in the TWAS, we didn't find any layer enrichment for those TWAS associated genes. That's not a big surprise, because we've been seeing quite a bit of differences between differentially expressed associated genes in schizophrenia and TWAS risk genes for schizophrenia. The results we presented today are based on the manual annotation of spots to layers using the spatial LIBD that Kristen explained earlier. However, in the future, as data sets get larger and larger, right, and so that means that they get harder to annotate, data-driven methods will be of use to all of us. So we teamed up with Stephanie Hicks and Lucas Weber from the Johns Hopkins Bloomberg School of Public Health Department of Biostatistics to investigate how these data-driven methods perform on our laminar data. You first need to identify genes of interest, which can be highly variable genes without using any spatial information, or spatially variable genes. You can then reduce the dimensionality of the data and perform spot-level clustering. This can be done sample by sample, or using all of the samples at the same time. And you can also use information from the image itself, such as the X and Y coordinates of the spots. One recent method is called spatial DE by Svensson et al. And this method identifies genes that have spatial patterns. However, even if you use this method, it's best to inspect your data, as spatial DE might identify genes that, yes, they have a spatial pattern, but it might not be the pattern that you're interested in. For example, here, MLBP, PCP4, and SNAP25 are all genes that are laminar genes that we expected to see. However, we also find genes such as HBB, IGKC, and NPY that, yes, they do have laminar patterns, but they're not informative for us. And these 3 genes are actually associated with blood flow. So a more global comparison we did was where we compared the spatial DE statistics on the y-axis against our ANOVA model F statistics on the x-axis. And so this plot over here shows in yellow, in different tones of yellow, a few genes that agree in between the two methods. And so these actually correspond to MOBP, PCB4, and SNAP25 from the previous slide. However, we also have some genes in red that have a low statistic on our x-axis, which is from our model, but have a high value on the y-axis. So these are the three blood flow related genes from the previous slide. However, you won't always have the x-axis because that depends on having a manual annotation of your spots. So you might not always have a method that you can compare like this. Stephanie and Lucas derived many clusters, some of which are completely unsupervised. Some of them are semi-supervised in the sense that they use the data-driven marker genes from Kristen's manual annotation. And these semi-supervised will become more prevalent as more of these data sets are shared among researchers, right? Because you can imagine like someone annotates the spots on a set of samples, let's say on a set of VLPFC samples, someone else generates a second data set of VLPFC samples. They can use the results from that first study to try to use a data-driven approach for annotating their spots on the second study. Or you can also use known marker genes from previous studies. However, known marker genes might not necessarily be expressed in the organism and the biological conditions that you're studying. To compare the clusters against the ground truth from our manual annotation, we use the adjusted rand index shown on the y-axis on these plots. And so here the clusters were derived from either PCA or UMAP, PCA shown on the first row, UMAP shown on the second row, dimension reduction techniques for either the spatial DE genes, spatial DE genes for the given image shown in blue, the spatial DE genes for all images shown in green, the highly variable genes that do not use spatial information shown in red, or our semi-supervised approach shown here in orange, or the known marker genes shown in purple. For each of these methods, we used the spatial coordinates shown here as the darker colors or x's. And we also tried it without using the x and y spatial coordinates shown in light colors or circles here. And so overall, like there's a lot of methods shown in this slide, right? And we also included here the p-value showing the comparison between using and not using the x and y spatial coordinate information. And so for a lot of the pairs, so for example here, these two orange pairs, using the x and y image information did improve the results. For some of the pairs it didn't. But something else that was striking to us was that using the highly variable genes that don't use actual spatial information seem to perform better than the spatial DE results. And so actually all of those cluster results, you can explore them through BB browser and that you'll be able to access through BioTuring. Now, given that we shared our data at the time that we posted our preprint in February of 2020, other researchers have been able to test their new clustering methods with our data. And that's because our data is a more complex tissue than the example mouse data shared by 10x Genomics. So shown here are results using base space by Zhao et al., which outperform the methods we used and showed in our paper. And this particular method-based space is now available through Bioconductor. They actually use the same adjusted rand index metric that we used. And so you can see over here that base space perform pretty well. And if you compare the manual annotation from panel A versus the annotation they obtain over here, it looks strikingly similar, right? Like visually, as the visual inspection. Our team has also been involved in the effort to standardize open source R bioconductor objects for handling spatially resolved transcriptomics data, including data from Visium. And so this is work in collaboration with Dario Rigelli, Lucas, who's part of our team, and Elena Crowell, and it's all being open source, and it's available now through bioRxiv. We have also been working on our spatial LIBD R bioconductor package to make it easier to interactively explore new Visium datasets. This is work led by Brenda Pardo and Abby Spangler, and we're hoping to make it available very soon on bioRxiv too. In summary, we use the 10x Genomics Visium platform to investigate the spatial patterns of gene expression in the human DLPFC. We annotated each of the 55 micron spots to layers using our web interactive application that we built for this task. We identified new layer-specific marker genes and validated them experimentally. Using this data, we can spatially register cell or nucleate clusters, as well as investigate the layer enrichment of genes associated with disorders such as schizophrenia and autism spectrum disorder. Our data and tools are documented and available from research.libd.org/spatial LIBD. And our results are described in our Nature Neuroscience paper from this year. This project was truly a team science project involving data scientists and biologists across LIBD, Johns Hopkins, and 10x Genomics. As you can see, we have a lot of ideas and future work. And if you're interested in joining our teams, please let us know. Thank you very much, and we'll be happy to answer as best as possible any questions you might have.
Thanks, Leo. Thanks, Kristen, for your sharing. Next up, Tree, our head of software development, will be sharing with you guys on how you can interactively access the data set of Leo and Kristen in bbrowser. Tree, you can share the screen now.
Yes, I just share my screen. Hello, everyone, and thank you, Kristen and Leo, for really interesting papers. So if you want to access the data behind this paper, you can, of course, go to the link that Leo provided in his slides, or using the spatial Lieber package, where you can access the data using our programming interface. For user with bbrowser, if you don't have bbrowser, you can go to our website and download. This is a desktop application where we help you to connect with our single cell database. And the interface of bbrowser is very simple. When you open, this is look like a database where you can query for data set. So to look for this data set, you can just type in any keyword that related. For example, I type here is the prefrontal cortex. So you will find the study at the second items, and there will be several information, including the title, abstract, and also information on the data itself. So this is a Visium data set that includes two types of information. One is the expression, and secondly is the spatial information. So this is one of the really special data that we actually have to update our software as soon as possible so that it can adapt with this new kind of data structure. So at first, when you enter this data set, it will show you a TSNE consisting of 47,000 data spots. So each data point here represents a data spot on the layer, and this is coming from 12 different slides over here. And on your left-hand side, you can see there is a series of different slides where whenever you enter into a subset of this data set, it will show you not just the TSNE of the data, but also the slide as well. So this is a study that consisting of multiple slides On the other hand, you can visit the metadata. By default, whenever you visit a subcluster, it will try to render coloring the cell using the default metadata. This is the graph-based cluster. You can, of course, visit other metadata that Leo has provided us as well. So he includes almost every metadata that he mentioned in the papers, including first is the label about the layers that he uses layer enrichment method to classify. Other kind of metadata can be also very useful, not just about categorical, but also numerical. For example, the ratio of mitochondrial genes. So this is an interesting metadata where you can actually see the ratio of mitochondrial gene on every data spot, and it somehow have you to estimate how the mitochondrial gene expression distribute differently in different layers. So one way for you to further explore the data is by looking at the composition of cell under different level of mitochondrial ratio. So one way for doing that is that you can play around with the box selection in this graph, where you can select a set of cells. For example, I select a group of cells that have very low ratio mitochondria over here. You can extend the composition plot. And right now, this composition plot will try to break down what you're selecting using a secondary metadata. So as you changing your mitochondrial ratio range, you will see the composition of different layers changing from white matters to more of the outer layers of the tissues. Right. So one other very interesting metadata that you would also like to visit as well is the count. So I think the author put in here the number of nuclei count underlying behind the data spot. So by default, because of our quality control team, they try to verify the results. So right now we are viewing it as a categorical. You can go to edit and definitely can change any metadata to numerical, just like this. And so for this kind of metadata, you can see there are some cells that have very low number of cell count. And this can be observed further by increasing the resolution, where we can only look at the data spot that only have 0 or 1 cell count. And for spatial data, you will have the spatial image at 2 different resolutions. You can change to higher resolution. So in this case, we have the blue data spot indicate for the data that have no cell body or the neutrophils mentioned in the papers. So in the high resolution mode, you can actually zoom in and then lower the resolution of the point so that you can actually see what is behind those data spots. So that is another way that you can play with the spatial tool that we provide. So it's not just about seeing and viewing, you actually can create your own annotation on this kind of metadata as well. So let's go back to layers. Right, so one important marker that I think that mentioned also in the paper is the keratin 17, which indicate for the markers of layers number 6. So with this software, you can query a gene, 2 genes, or multiple genes at a time, and it will provide you with multiple ways to support your visualization. So right now, my metadata is in layer reorder. So which means on the x-axis of this volcano will be the layers that I'm seeing on the active metadata. So if you want to, for example, get rid of the NA group, you can just uncheck the box and then observe this marker for layer 6 over here. So not just about seeing the reproduce what the author found in their paper, you can also further explore the data set. So we support several tools, not just observing the composition, but also finding markers as well. So for example, I'm selecting layer 6 over here, which is highlight at white data spot on the screen, I can further extend the marker features and click on find markers. So what I'm doing right now is actually a differential expression analysis that compare layer number 6 to the rest of the data that is visible on the screen, which is all the layer except the NA layers. So if you sort the marker result based on log phone chain, you can easily find that keratin 17 is right now on the top of the list, right? And also along with other types of markers. So if you want to get rid of, for example, Y matter, because this is very different to other layers, which can create a lot of bias and introduce some not very significant markers, you can just uncheck it and then rerun the marker gene. So right now we can still see keratin 17 on the list, but other than that, we will get another set of genes that also indicate for the enrichment or in layers beside keratin 17. And whenever you query for genes, we link that ensemble ID to our own genes database, where we can tell you the function of the genes and where which gene ontology or function that the gene enrich. Okay, so one last very interesting I think analysis that you can done with B-browser is that this is not just a software where you can look at one data set at a time. So because we are linked this tool with one of the largest single cell database right now, we have around 20 million cells. We can actually perform many more exciting meta-analysis at database scale. So what I'm trying to do show here is that I'm selecting a group of cells in layers and then click on cell search. So what cell search does is not a cell deconvolution method. It's just basically getting an expression profile out of the cell that I'm select. That can be any kind of selection. That can be, so right now I'm selecting based on layer number 6. So you can see here that it tells me that my selection match 2 different populations in the database. And as will show here the information of the papers, and also the type of the type of the cell that this paper label the similar cell type. So we can see here we have 2 hits. The first hit is a group of cell in this paper on brain, and this is a group of excitatory neuron. So on the second hit, we have a group of another type of cell. This is oligodendrocyte from another study. So if you click on study composition, you can actually further break down what are those cells. So for example, I can go to the metadata of this paper and click on, for example, donors. So I will know that most of the oligodendrocyte coming from donor 52. And if you're interested in this paper, you can always click on explore this study. And this will bring you to the front page where we search for the exact study and you can download and further explore. So that is basically I want to introduce. There is actually many ways that you can explore further analyze papers in B browser. And I think for now, let's stop here and then we can answer any question that you might have. But if you want to access the data, again, you can visit the link from Leo slides, which we will send after the talk. And also, you can always visit B browser and search for this paper. I hope you find this tool useful, and thank you.
Thanks, Tri. Again, thanks, Leo. Thanks, Kristen and Tri for the talk. And it's time for the Q&A section. I see that there are a lot of questions. Some of them have been answered by Kristen and Leo. I will go through them one by one. So the first question is that, are there any white papers describing the VISM immunofluorescent protocol? So Kristen has already copied the link to the white paper into the Q&A box so that you guys can check it out. The second question, Yoon sent it to the chat box, is that Yun is not clear about the layer means. So Leo has also typed in the answer into the chat box, which you can also check out. And another question is from Yun as well. For IHC, do you need standing layer neuron marker to show layers? So Kristen or Leo, can you answer this?
I can take that one. So unfortunately for the IHC like approach, like you don't get the histology. So it's kind of a choice. Either you do the immunofluorescence or you would do like the H&E approach. So if it's like important to you to know where all the cell bodies are, then you need to have some kind of stain incorporated in your antibody panel. We also include DAPI. Sometimes that's enough based on cell density to help you figure out, you know, where your different regions are. But yeah, anything you'd want to use for annotation of your spatial regions, you would want to incorporate into your antibody panel.
Yep, thanks, Kristen. Another question is from Greg. What are your thoughts on filtering out spots with high mito content in spatial datasets versus single cell datasets? I have been playing with different datasets from the mouse brain, and the percentage of mito genes per spot is much higher than what one normally sees in single cell dataset. How did you deal with this?
Yeah, no, I'll take that question. So there's several things to consider. First is that in our particular data, we noticed that the spots that had high mitochondrial percent of reads were actually related to layers. So they were not like random spots. They had a specific spatial location. So this means that it was like a biological difference rather than a technical one. So for our data, we didn't filter spots based on those metrics. However, like we've seen other data sets, and so sometimes you can find random spots that have maybe low quality data as detected using this mitochondrial mapping percent, and you might want to consider filtering them out. However, I think like as a field, like we need to tweak what are the quality control steps that we used to use for single cell or single nucleus data, and then adapt them for these the properties of Visium data. Kristen also was telling me, we were chatting, and so Kristen told me like something else I was forgetting, which is with Visium, you're actually capturing, you know, the full, you might be capturing more than one cell, right, in a given spot, and you're actually capturing the mitochondria there. With single nucleus or single cell RNA-seq methods, you might be just capturing what's part of the cell. So you might be removing the mitochondria, sorry. So this could lead to a higher chromosome M expression percent in Visium data compared to single nucleus or single cell RNA-seq data. So both of these things are things you might need to take into account, and it means that you might actually need to have more reads with Visium data, right? Because you can expect a higher percent of them to be aligned to the mitochondrial chromosome, right? And so you might need more reads because then you want to have more genes measured, right? So all of these things are things that we're learning. There's no specific guidelines yet.
Yeah, thanks, Leo. Another question, is it possible to install it locally due to restrictions in data uploading into cloud? I guess this is a question for bbrowser, so Tree will be answering this.
Thank you for the question. I think right now, bbrowser have two modes, which is it can either run on the local one, which is by default when you download from our website, is that everything from data analysis to storage will be done on your local machine. So when you access a public data set, it's actually download that data set to your computer, and all of the data analysis and visualization will happen right on your laptop. And the other mode is for enterprise version of the software where you actually can deploy the analysis module on a separate physical or a cloud server, and your laptop right now will act like a thin client where you only handle a limited numbers of data, just enough for visualization.
Okay, thanks, Tree. Another question is that, do you plan to develop Visium for nanostring data visualization?
I can take that one. Was that for me?
I guess this is for, I think for both Kristen and Tree. Kristen, you can answer first, and maybe Tree can add in.
I was just going to say, on the data generation side, we're actually very interested to kind of work with long read sequencing data from these libraries. So this is something we're actively collaborating on and trying to work out, but not yet close for prime time, but something on our radar.
Yeah, thanks, Kristen. And on the software side, Tree, you have any comments on this question?
Yeah, so we actually trying both. So we have right now we have the module to log Visium data, and we also working on another module to support nanostring as well, because we receive a really high demands from our current customer on using the same tool for their nanostring in-house. But if you have any like a sample or a specific case study that you want to escalate the development, feel free to contact us on the support@bioturing.com so that we can like contact you and update to you the development in real time.
Okay, another question. When perform H&E staining, is that possible to separately perform IF staining and realign the IF image with H&E image or ST data? So Kristen or Leo, can you answer this question?
So yes, technically, I guess it depends what you're staining for. So we've tried this to take like adjacent tissue sections and stain for even different antibody panels to maximize the number of antibodies to look at. It really depends how big your structures are and if they would span into two different sections. So for larger kinds of neuropathology, this might work. One challenge is picking up tissue sections really precisely so that you don't have any folding or tearing, because this can make it very difficult to align two different tissue sections. So it is possible, and there's some studies out there where people have tried to do this on the older platform, the spatial transcriptomics platform, before Visium was developed. So you could check their kind of methods to see how they did this, but it's definitely more advantageous to do it in the same tissue section.
Yeah, thanks, Kristen. Another question, could B-browser show multiple slices side by side and users can explore interactively across them?
Yes, that is a great question. So it's actually what is on the top list of our to-do list right now. So we're working on functionality where you can stay at the main cluster and view all of the subset at the same time. So that way it will work for this particular study when you stay on 47,000 data spot and you will see all of the 12 slides. So this is one of the things that we are in development, and I think it will be available in 1 month or less.
Yeah, thanks, Tree. Another question in the chat box from Yoon. As we know, we only use one slide with 4 sections for spatial gene expression. For IHC, how many slides you suggest collect? So Kristen, can you?
All right, can you repeat the question?
Yeah,
I was just looking
in the chat box. So as we know, we only use one slide with 4 sections for spatial gene expression. For IHC, how many slides you suggest to collect?
It really depends on what your biological question is. I mean, we found in our in our study where we did these spatial, replicates, the adjacent spatial replicates, that there was very, high correlation between the two sections. So we've moved away from doing like the adjacent technical replicates in favor of doing more biological replicates. So this really just depends on what your kind of biological questions are, and how many antibodies you want to try and multiplex with, and what kind of imaging system like you have in-house that will support that level of multiplexing, if that makes sense.
Yeah, thanks, Kristen. Because the clock is ticking and we still have a lot of questions, I'll take them. The final question, which is, what is the underlying method for the cell search? So Tree, can you answer this question?
Oh, I think this is an excellent question. So this is one of the really the most difficult thing that we have to overcome when we try to perform meta-analysis on at database scale. So the problem of the cell search that you can clearly see is that different data set in our database will be generated and analyzed under different parameters and methods. So there's a lot of batch effect there. So we are working on writing a full description in the papers where we can clearly explain how we perform the cell search. But I can summarize for you briefly is that we have to index all of the cell type and cluster base in our database into a single profile. And then given a set of cells that you select on the screen, we can also generate a single profile using the same method. And then we will compare the methods using some of the non-parametric methods so that we can overcome the batch effect. So it really requires the database has to be indexed, and also the assumption on the way you're selecting the cell, right? So we assume that when the user select a group of cells on the screen, they mostly coming from the same tissue, the same layers, or the same type of cell. So we assume some of the homogeneous attributes right there in your selection. And that is how the method is developed at the moment.
Okay, thanks, Tree. So I think we are running out of time, although we still have some questions in the list. But once again, I would like to thank everyone for joining, especially thanks Kristen, Leo, and Tree for delivering wonderful talks and for answering so many questions we have today. I think we have a great discussion, and I hope everybody enjoy today's webinar, and hope you guys have a nice day. See you next time on our upcoming webinars.